experience in research field. His specialization area is genetics and biotechnology. And he had a very great achievement. He worked as a postdoctoral fellow at National Research Center on Plant Biotechnology, IARI, New Delhi, on genetic engineering and gene primordial in BT Brinjal. He worked as a scientist in well-known private seed companies working on cloning strategies of biotic and abiotic genes, BT protein qualification, and zygosity. He is a member of and POS in botany for undergraduate course and general warden new PG hostel Usmania University. He had completed a UGC BSR research project on linseed and working on another project sponsored by UPE FAROU. 6 research scholars are awarded PhD under Sir guidance and some more are in pipeline. He has written 2 book chapters published in well-known publisher Willie VCH publication Winham and authored first year BSc Botany textbook of Telugu Academy. Besides he published 45 general papers in national and international journals with high impact factor. He is a life member of different societies of science academies, attended several national and international conferences, presented so many posters in many symposia, workshops, and delivered many lectures. So, Kiran, I welcome you to this webinar. Today's topic is production of phytochemicals through tissue culture technique. Thank you, Kiran.
At the outset,
1 second, Latha madam want to speak.
Sure.
Good afternoon, everyone. Respected patron in chief of this webinar, Dr. B. Sunita Padmavati Garu.
Voice is not audible, ma'am.
Okay.
Voice not.
1 minute. Good afternoon, everyone. Good afternoon, everyone. Respected patron in chief of this webinar, Dr. B. Sunita Padmavati Garu, Principal of Govt.Degree College for Women, Hussainialam. I cordially welcome our distinguished resource person, Dr. B. Kiran Kumar Garu, Assistant Professor, Department of Botany, Usmania University. I also welcome to all who are participating in this webinar. My dear students, Department of Botany organizing this webinar on production of phytochemicals through tissue culture technique. All of you know about the drugs produced by the plants and herbs. Medicines have been used since ancient times to treat human and animal diseases. And at present, several countries still depend on herbs and plants as the main source of drug. So plants are also considered as a biofactories for the production of phytochemicals. Phytomedicines have emerged as a cost-effective, eco-friendly and better alternative of synthetic medicines. Tissue culture technology is now considered as a highly efficient tool for the production of such phytochemicals under the controlled condition. Plants are a rich source of the various secondary metabolites. So today Department of Botany organizing this extension lecture on production of phytochemicals through tissue culture techniques. This topic is very important, ma. Please listen carefully. If you have any doubts, ask questions after the lecture session. And a special thank you to our principal madam for giving permission to conduct a webinar. And also I thank you very much for our resource person, Dr. B. Kiran Kumar Garu. In spite of his busy schedule, he has accepted and to give lecture of this webinar as a resource person. Now I hand over to mic to our resource person. Please continue lecture session, sir.
Thank you, Latha ma'am. At the outset, to everyone, to Sunita ma'am, to the principal ma'am, Dr. D. Kiran Guru from the Department of Botany. Already spoke about my academy. Today I'm going to talk on the production of phytochemicals through tissue culture techniques. All well aware of technologies, the tissue culture technique will going to. Can my PPT is visible?
I'm now coming, sir. Yes, sir.
Yes, sir. Yes, sir.
Before I'll start with the actual phytochemicals. I think the students are well aware of these secondary metabolites. Yes, you know about what do you mean by secondary metabolites and how these secondary metabolites will take a role in plant growth and development. Okay. So actually, the secondary metabolites does not play any role in the primary plant and growth development, but they play an important role in the plant defense mechanisms. Okay. So as everybody know about how the plants which are there at a particular place, which cannot move from one place to another place, how they are going to defend from biotic and abiotic stress. Anybody anytime think about that? So since human beings, if you feed every child, you go and wrap the blanket, all those things. But plants cannot move from that particular place. How they are going to face the environmental stresses? Then these secondary metabolites comes into the picture. The secondary metabolites, which are normally these are the organic compounds, okay, which are not directly involved in the primary growth, development, or reproduction of the plant, but they play an important role in the defense mechanisms. Okay? For example, they used to fight from herbivores, pests, and pathogens. These are different types of biotic and abiotic stresses the plants has to cope up. Okay, so there are so many phytochemicals which are being produced in the plants, which are so much potent, which are very much potent, and about 1500 new molecules are reported from the plants each year. Okay? From the each year, up to 30%, and out of this, all the phytochemicals which are being explored, they are being having some biological activity. Okay? They show about all the compounds may not show all the biological activity. Some of the compounds may show some antibacterial activity, some show antifungal activity, some are antioxidant, okay, all those things like that. So, in humans or plants, secondary metabolites are of great importance. Okay? You can use nowadays all these secondary metabolites which are produced. Plants are generally used in the medicine. Sometimes they used as a cosmetics, additives, dyes, antimicrobial agents, okay, etc. They have a very vast application in the various industries. So, the secondary metabolites is linked to the induction of morphological differentiation. Okay? tell you what is morphological differentiation when I come to the actual topic of tissue culture. There are two types of differentiations. You can see that in the tissue culture, like redifferentiation and dedifferentiation. Okay? So since you know about the beauty of the plant system is the plant cells are totipotent. They are totipotent. Totipotent in a sense, uh, from a single cell, the whole plant body is makeup. Okay, as such, they are totipotent. The presence of this totipotency in the plant systems, the particular plant kingdom, makes that particular all are joining late. So this totipotency as a big boon is a big boon for the plant tissue culture. The efficiency of a single cell transformed into the whole plant makes it unique. Okay, so that's why we develop whatever the phytochemicals which are present in the plants can be linked to the induction by means of a technique called as a plant tissue culture where morphological differentiation will happen. Okay, so for that things, cultures has to be initiated by simply fetching whatever the cut sections from the surface sterilized plant organs on a nutrient medium containing the suitable phytohormones. You have already studied about the phytohormones. There are different types of phytohormones like gibberellins, auxins, okay, ethylene, all those so many. There are some inhibitors as well as the growth regulators. Those are called as the phytohormones. So whatever if you cut any plant, later on if you want to produce the callus, so before that you need to surface sterilize the explant. Explant is nothing but explant is anything which is any part of a plant, whether it is a toddler node or it is whether it is a leaf or whether it is a tip, okay, root tip or whatever it may be, whatever the part which you are going to use in the tissue culture, we call that as an explant. As such, you cannot keep the explant directly onto the media. Okay, since there is a contamination, you need to sterilize the explant before placing them onto the tissue culture medium. That we call it as a surface sterilization. Okay, that is done by the chemicals like mercury chloride, sodium hypochlorite, all those things. So once you do the surface sterilization of that particular explant, if you introduce that explant on a medium containing suitable hormones, suitability of the hormones is also plays an important role. All the hormones which you are going to subject them to the medium, you need to do the standardization before. Okay, all the plants will not respond in a same way. Okay, all the plants will not respond in the same way. That's why you need to do the standardization of a particular plant before growing them in a tissue culture. Okay, so generally the auxins and cytokinins are generally used for this purpose. And on such a medium containing exhibit callousing, which usually starts at the ends and gradually extends over the entire surface of the tissue. Whatever the explant which you have kept on the nutrient media, okay, after a few days means after 10 days or 2 weeks, okay, whatever the cells which are in contact with the some undifferentiated mass of a cells, okay, they exhibit undifferentiated mass of a cells, which we call it as a callus. Okay, callus is undifferentiated mass of a cells. Okay, so this how much quantity of auxins and cytokinin needs to be incorporated into the medium. All these things needs to be will be done by the standardization of protocol. We need to do some standardization protocol where how much of media is, how much of phytohormones are need to be added to that particular media. Okay, normally in plant tissue culture, you use the Murishige food media, which generally called as a MS medium. Okay, MS is nothing but Murishige food medium. It is almost generally used plant tissue culture media, which is generally used around the world. So, coming to the types of secondary metabolites. Secondary metabolites are nothing but they are the phytochemicals, right? Okay, there are three types of secondary metabolites. We have studied, I think. First group terpenes. Second one is the phenolic compounds, and the third one is the nitrogen-containing compounds. Okay, these are the three classes of secondary metabolites where they play an important role in the defense mechanism of the plants. Apart from that, the plant secondary metabolites have different applications. For example, if you see the defense mechanisms, there they plays an important role in the insect, molluscs, and vertebrates. In case of herbivores, apart from that, they are antimicrobial, antiviral, antibacterial, and antifungal. Apart from that, they also plays an important role in the inhibition of germination and secondary and growth of the seedlings, inhibition toxicity, repellence, deterrence, toxicity, growth. All these are comes as a defense responses to a particular plant. Okay. Apart from that, they also attract some pollinating agents like insects. Okay. Dispersals, animals, root nodules, bacteria adapt to herbivores. You know about the root nodulation of rhizobacteria. Okay, in the plants of Fabaceae members, in the root nodules. That is also that is the production of plant secondary metabolites, which plays an important role in the induction of those root nodules in the Fabaceae members. Apart from that, they also produce, they also protect the plants from UV protection. Okay, all these things, the secondary metabolites plays an important role in the plant defense mechanism. So there does not play any primary role, but all these the secondary roles are very important for the survival of the plant. So how we are going to produce the phytochemicals from the tissue culture? The phytochemicals from the tissue culture can be produced in three ways. The first one is called as a cell suspension culture. Second one is called as a hairy root culture. And third one is called as a immobilized cell culture. We are going to learn individually in detail. Okay. So before we'll come to the actual three types of production. So what are the advantages of this production of secondary metabolites through tissue culture? Okay. So what are the advantages? Compounds which are produced under controlled conditions. Okay. They are controlled conditions as per the market demands. That is the first advantage. They are independent of environmental factors, seasonal variations, microbial diseases, geographical variations, etc. Culture systems are independent of environmental control. Whatever the compounds which are produced in the tissue culture, they are environmental friendly. Whatever the conditions which you are going to provide in the tissue culture by make those lot of disturbance. All the people are joining now. So all the whatever the environmental factors which are going to provide in the environment in the plant tissue culture, they are make use of those controlled conditions. Okay? They are make use of those controlled conditions and they do not depend on the environmental conditions which happens outside. So they are independent of those things. Apart from that, seasonal variations. Okay, there are some compounds which are produced in a particular season. Okay, in the plant systems. But here, if you are using the tissue culture, independent of a seasonal variation, they can be produced throughout the year. That is also one of the beauty. Apart from that, pest microbial diseases. The plant is existing outside. There are so many biological stresses in the form of diseases, the pathogens, or the pests which going to attack on the plants. But in case of tissue culture, since they grow in vitro and controlled conditions, we we are not we are least bothered about the pest and microbial diseases. Apart from that, geographical variations. Geographical variations also plays an important role. Some of the plants which are growing in the some other North India may not flower in South India. If you take the same plant, take that is geographical boundaries. So even the tissue culture plays an important role. It is free from geographical variations. Okay, these are the disadvantages where the phytochemicals can be produced through plant tissue culture. And apart from that, cell growth can be controlled to provide improved product formation. You can control the cell growth. So if you want that compound to be produced for a minimum time, you can do that. Once it is over, you can switch off the system. Okay, that is nothing but the controlled conditions to provide the uh, phytochemicals. Quality of the products will be consistent. That is, it remains same as it is produced from a specific cell lines. Whatever the specific cell lines which you are using, okay, the quality is similar. You cannot face any adversity in the products. And apart from that, recovery of the product will be very easy. Okay, the product is formed in the tissue culture, recovery of that product from the tissue culture is very easy. Okay? That is how they plays an important advantages. Apart from that, plant cultures are particularly useful in case of plants which are difficult to grow in the flames. Another important aspect is whatever the phytochemicals which cannot be produced in the fields, can be produced in the tissue culture. That is also one of the important aspects. And apart from that, mutant cell lines can be developed for the production of novel compounds of commercial importance which are normally not found in the plants. So, here, have you heard about any time about the mutation? Mutation, you know about the sudden heritable changes which happens. Okay, whatever the mutations, if you want the a particular plant, if you mutate that particular plant will produce the phytochemical in a more quantity. Okay, in that particular case, you can cause mutations by either the physical mutagens or the chemical mutagens, and you can produce that compound in a very high amount. Okay, those lines we call it as a mutant cell lines. Okay, so apart from that, production time is less and labor costs are minimal. This is very important. Whatever the production time which is very less and labor cost is also very minimal. These are very important criteria. Okay. Apart from that, 25 to 30% of the medicines for human use and various chemical materials are obtained from the plant tissue. Now it is nearly not 20 to 30. Now it is more than 50%. More than 50% of the medicines which generally human use and apart from human use or various chemical industries which we are using, they came from all the plant tissue culture techniques only. Okay, so all these are the advantages. So every technology has pros and cons. So now we are going to study about what are the cons. That is nothing but the disadvantages. Non-differentiation can produce little amount of compounds if there is a no differentiation. As I already told you in the initial, once the cell is whatever the cell or whatever the explant which you are going to grow them in a aseptic condition in a culture media, if that particular cell that does not differentiate, okay, it does not differentiate. How, why that does not differentiate? It is entirely depends on the standardization protocol. Whatever the auxins and gibberellins which you are going to use, if you are not using in a proper permutation and combinations, then this type of disadvantages will happen. And apart from that, culture cells are genetically unstable and may undergo sometimes mutations. So this is a normal thing which happens in the tissue culture. Okay, we call the mutations which happens in the tissue culture as a stomaclonal variations. Okay, those stomaclonal variations are also an important thing which we can use that variance for the production of secondary metabolites in a large scale. Okay. Apart from that, vigorous steering is necessary to prevent aggregation of the cells. So, during this process, whatever the processes which we are going to use, like cell suspension culture or immobilization or aerobic culture, we have to take a prevention of aggregation of cells. Okay? That is very, very important. If you cannot prevent aggregation of cells, what will happen due to the continuous rigorous stirring, the cells may get damaged. And apart from all these things, the main important aspect is aseptic conditions. If you don't maintain the aseptic conditions, the whole culture experiments will be contaminated. Okay? Since whatever the different types of bacteria, viruses which exist in the airborne, okay, if you don't maintain aseptic conditions, they will going to spoil the tissue culture techniques. Okay, all things will be get contaminated. If those things are contaminated, it is just you have to throw them in the dustbin. That's it. You cannot do anything. So that's why aseptic conditions is very, very important in the plant tissue culture. Any infection during the culture growth can adversely affect the production of secondary metabolites. That is also important. So whatever infections, in spite of providing 100% aseptic conditions, there are chances. Okay, if you are providing 99.9%, there is a 0.1% chance is also there that any infection may occur. Okay, in that particular case, the whole phytochemicals, whatever the production which is happening, it gets adversely affected. Okay, that is a different type of the different types of disadvantages. See the advantages and disadvantages. The advantages way more, okay, rather than disadvantages. If you take preliminary precautions of all these things, then the tissue culture production of phytochemicals is brings good returns. So these are the examples of plant metabolites which are produced by different plant cell cultures. Okay, I'll tell you about the different types of compounds. Chiconin. Chiconin is a secondary metabolite which is obtained from the plant Dithospermum erythroxyron. Okay? Dithospermum erythroxyron. Okay, the culture type, whatever the culture type which we are going to use is cell suspension. Inosinocyde, anthraquinones, admelycin, ubiquinone, diazoxanide, berberin, anthraquinone, all these are the different types of secondary metabolites are the phytochemicals which are being produced from the different types of plants. Okay? And what are the culture type? Culture type, you can able to see the SCS. Okay, S stands for cell suspension culture and C stands for callus suspension culture. What is the see the yield also, there is a vast difference. In the plant, it is 1.5. If you produce that in the culture media, it is 20. Okay? There is a vast difference in the production of the secondary metabolites in the normal conditions by the plants. And if you compare that with the plant tissue culture technique, okay, there is a vast difference in the production of the compounds. So all these are the different types of compounds that have been produced in a large scale using plant tissue culture. Now we are entering into the different types of tissue culture techniques. I told you about the cell suspension culture. This is one of the most important culture technique. No doubt it is based on the plant tissue culture, and most of the secondary metabolites are whatever the compounds which are being produced, they are being produced largely from this cell suspension culture. Okay? It is very convenient to handle, and it involves the various stages. The first stage, the cell suspension culture, is selection of suitable cell and callus formation. So, under that, what the first step is develop a callus culture from the whole plant. For this purpose, we select a cell having ability to generate the whole plant cells, which are called as the totipotent cells. I told you earlier, and undifferentiated mass of cells on the culture plate, we call that as a callus. Callus must be obtained from the totipotent dividing cells. So the beauty of the plant kingdom is entirely depends upon the totipotency. You take any cell of the plant that will going to give rise to the callus. Okay. If you see the cells of a humans, okay, they are very differ from the plant cells. Okay, the liver cells which are being produced in the human at the time of embryonic development, they only express in liver. Okay, heart cells in the heart only. But here, whatever the cells which are they are look alike and which are called as a totipotent cells, they gives rise to the whole plant body. That is the beauty of this plant tissue culture. Okay, due to the presence of this totipotency, so we are making use of that particular particular this activity of this totipotency for the plant tissue culture. Okay, not only in plant tissue culture, we can use this totipotency in development of genetically modified crops also. Whatever the genetical modified crops like BT cotton, BT brinjal, there are so many crops which are produced. They are being produced based upon this totipotency principle only. Okay. Apart from that, this totipotency principle also plays an important role in the production of secondary metabolites in very large amount. Okay, through tissue culture technique. Second stage is development of cell suspension culture. So here, the important factor count is culturing the cell in a precise, good, and sterilized medium. Okay, this is very important. Good and sterilized medium, whatever the medium which we are using. As I told you just now, most of the plant tissue culture media generally used is MS medium, Murashige and scoop medium. It is the most widely used media all over the world. A suspension culture is developed by transferring a small amount of callus into a liquid medium and is maintained under suitable condition of aeration, vegetation, light, temperature, and other physical factors. Once whatever the explant which you have kept on a neutral medium, from that once the undifferentiated mass of the cell, which we call it as the callus, develops, you remove some of the callus and you introduce this callus into the liquid medium. Okay, and you maintain this liquid medium containing by providing all the suitable conditions like aeration, vegetation, temperature, and other physical factors. If you maintain those things, then the cell will going to proliferate. As the cell proliferates, even the secondary metabolites which are being produced from that particular cell will also proliferate. Okay. That is the second aspect, that is development of suspension culture. Third, what are the media components? And media components are mechanism. Okay, so media components and mechanism plays an important, very important role in the production of a particular second molecular. Okay, cell suspension components includes fresh friable fragments of callus into 50 ml of Murashige and scoop liquid media in 250 ml Erlenmeyer flask. Apart from that, you are going to use regulators like kinetin, okay, 2,4-D, and their combination like benzyl adenine purine or with kinetin or naphthalene acetic acid. Okay, all these are the different types of phytohormones. Normally, the auxins plays an important role for the induction of callus. Okay, auxins plays an important role in the induction of the callus, particularly the 2,4-D and naphthalene acetic acid. If you grow anything, any explant under this phytohormonal combination, surely you'll get the cals. Okay. So apart from that, macronutrients and micronutrients, precursor feeding, exogenous supply of biosynthetic precursors, culture medium is applied to increase the yield of the desired product. Whatever the product, the phytochemicals which we are going to produce that in a large quantities, that whatever the media, which media components and the different combinations and permutations of these compounds, okay, like 2,4-D, benzyl and kinetin, all these things plays an important role. All these things need to be standardized before starting the actual experiment. Okay, all this plays an important role in the cell suspension culture. So apart from that, there is a process called as elicitation. Okay, elicitation is one of the important, most effective strategy for improving the productivity of the bioactive compounds. Okay, elicitors are nothing but elicitors are the signal triggering the formation of secondary metabolites. These are also molecules that stimulate the number of defense responses in the plants. Research over the past decade has focused on the mechanisms by which the plant cells perceive and transduce these biological signals to activate defense responses. So, there are, as I already told you, that the plants will going to face the harsh environmental conditions in the form of biotic and abiotic stresses. How they are going to cope up those harsh environmental conditions due to the production of secondary metabolites. These secondary metabolites production is felicitated by the technique called as a elicitors. Okay, so elicitors plays an important role in the plant defense mechanism where they cope up the biotic and abiotic stresses which happens outside. So elicitation is also very important where you can use this technique for the production of phytochemicals using tissue culture. Agitation and suspension culture is incubate on rotary shaker for agitation under dark in the growth room. Once you, whatever this callus which you have incorporated into the 50 ml of the liquid medium, Erlenmeyer flask, that needs to be agitated. Agitated in a sense, we use the shakers. Okay, we have shakers where you keep that and you need to provide the different how many type rotation per minute and the required temperature, whatever the required temperature which the particular tissue grows. If you provide all those things, that could be done in the dark conditions. Okay, why we'll do the dark condition is there is a every chances of sensitive issues which may arise. We don't know what are the phytochemicals which may be some may be light sensitive. Okay, in that particular place we need to grow them in a dark conditions. Each treatment consists of 4 completely randomized replicates without subsamples and growth assessments, which are performed by recording data every 5 days during 30 period for fresh weight. So once you collate this particular callus into the cell suspension culture, after 30 days you need to weigh the cell fresh weight. Okay, whenever you weigh the cell fresh weight, you can able to estimate the amount of phytochemicals which are also be continues to multiply through the cell suspension culture. These are the different types of elicitors which are generally used in the industry. Okay, plant species and product. I'll give a good example of taxus. The second one, Taxus canadensis, is a gymnosperm, okay, which produces a toxin called as taxol. The main important we are talking about. I'm telling you that taxol is the toxin. So you can ask the question how this toxin is helpful for us. Taxol is generally used for the anti-cancerous activity. Okay, this is the only gymnosperm which produces the anti-cancerous agent taxol, that too from this Taxus species, Taxus canadensis. Okay, so in generally that is produced if you use the elicitor like N-acetyl phytohexose. This, if you use this elicitor, whatever the explant which you have taken from the Taxus species, okay, if you use these particular elicitors, the taxol will be produced more in the plant tissue culture technique. Okay, the amount of production in the plant outside the lab and the amount of production inside the tissue culture varies. Okay, so if you want to produce taxol in a more higher amount, then you can use these elicitors. Okay, that is a good example where that is used as an anti-cancerous agent. Apart from that, there are so many types of penicillins. Phytozone, which are generally used for isoflavonoids. Okay, saponins can be produced from the oligogalactolinous acid, ethyl jasmonate, yeast elicitors, polysaccharides. All these are the different types of elicitors and their products. So these are the different the steps which are involved in the production of phytochemicals. Okay, here they have given an example like Dithospermum erythrozydon. Okay, it's a natural medicine. Okay, there they are taking ing leaves. They cut the ing leaves. They surface sterilize the ing leaves. Then they inoculate on a MS medium containing different types of combinations and permutation of phytohormones. Once that is done, then you can able to see the callus. Once the callus is formed, thus callus is inoculated into the liquid medium. Okay? That we call it as a cell suspension culture. Once the cell suspension culture is there, that can be, if you want to produce that in a very larger amount, okay, then they can be introduced into the bioreactors. Okay, cell cultures in the bioreactors. Then once the if you introduce that in the bioreactors by providing all the necessary conditions, then the cells will going to proliferate. As the cell proliferate, even the compound which is present in those cells will also be produced in a large quantities. They are being subjected to the different types of filtrations. They are nothing but downstream processes. Okay, they are being filtered by different types like HPLC, GC, all those things. Where can obtain a pure compound. Okay, that we call it as a natural medicine. This is the whole steps which are involved in the production of phytochemicals. Okay. So all measures needs to be taken care. like appropriate nutrients, concentration, factors. Okay, these are plays an important role, and if you take care of these things, you can enhance the yield and productivity of metabolites in the plant cells. Optimum aeration, agitation conditions, capacity of oxygen supply, intensity of hydrodynamic stresses, plant cells, control of temperature, pH, nutrient concentration inside the bioreactor, and apart from that, aggregation. I told you, aggregation cells is also important factor which needs to be taken care. Otherwise, the cells damage may happen. Whenever the cell damage may happen, then ultimately the phytochemical production will also be lowered. Maintenance of aseptic conditions for relatively longer cultivation period. All these things plays an important role in the cell suspension culture. The most favored all the industries for the production of phytochemicals. All these are the different types of advantages of suspension culture. I told you just now. Next comes the hairy root culture. Okay, this is the second strategy of production of phytochemicals. Hairy root culture. So, hairy root culture is produced by the bacterium called as agrobacterium, right? Agrobacterium tumefaciens harbors the Ti plasmid, whereas Agrobacterium rhizogens harbors the Ra plasmid. Ra plasmid is inducing plasmid, and Ta plasmid is tumor inducing plasmid. Okay, so this bacterium, which is a gram-negative bacteria, which is present in almost all the soils, once they infect the plants, okay, depending upon the whether it is being infected by the Agrobacterium rhizogens or whether it is infected by the Agrobacterium tumefaciens. Okay, if they infected by the Agrobacterium tumefaciens, that results in the formation of crown gall disease. Okay, crown gall disease is nothing but whatever the tumor-inducing genes which are present on the plasmid, those are being transferred into the plant genome. Once it is incorporated into the plant genome as a part of its transcription process that also synthesizes the genes which are responsible for the formation of tumors. Okay, that is how the crown gall disease is formed when it is infected by the Agrobacterium tumefaciens. If anything, other plant which is infected by the Agrobacterium rhizogens, it contains the Ra plasmids, okay, root-inducing plasmids, which plays an important role. There are some phytochemicals which are produced in the root system rather than shoot system. Okay, so there are some compounds which are produced system rather than shoot system. Making use of this hairy root culture, we can produce the phytochemicals which are confined, which are produced in root system. Okay, if you treat the Agrobacterium rhizogens, you can produce the large amount of compounds which are produced in the root system. This is the Ra plasmid. Okay, Ra plasmid, you know about this plasmid, you know about the plasmid, right? Okay, plasmid is the extra chromosomal segment, okay, self-replication. So there you can have this NPT left border and right border, whatever it is in between the left border and right border will go and incorporate into the plant gene. Okay? You know about every gene has a promoter, terminator, and this thing. Okay, that is how the transcription starts. You know about the central dogma of molecular biology. DNA to RNA to protein. That is whole the universe is entirely depend on this central dogma. Okay, so Ra plasmid plays an important role in the production of hairy root. Okay, whatever the compounds which are being produced in the system, we make use of this Agrobacterium rhizogenes bacteria. Okay, this is the Agrobacterium cell and plant cell. How this T, that is tumor inducing or stand gets incorporated into the plant genome. You know about the there is a transferring of genes from prokaryotes to the eukaryotes. This bacteria is a prokaryote and plant cell is a eukaryotic system. Okay, you can understand the tDNA which is transferred from the prokaryote to the eukaryotic DNA. There is every chances of defense mechanism which exists in the eukaryotic system. Whenever the plant outside DNA enters, there are some endonucleases which will chop down the DNA which enters into the plant cell. So there is some mechanism which is being provided by the Vir genes we call it as. Okay, they forms a tDNA complex where whatever the DNA which enters from the bacteria into the plant cell is guarded by that particular T complex against the endonucleases. Otherwise, the tDNA once enters into the agrobacterium into the plant cell, the endonucleases which are present in the plant cell will chop out the DNA. Okay, so that is the beauty of this agrobacterium. Okay, all these virgins are present on the tDNA stand itself. Okay, so that's why the production of genetically modified crops by means of agrobacterium tumefaciens opened new avenues where the BT cotton is one of the important cotton crop all the world that plays an important role. Okay, you know about the cotton plants. Okay, cotton generally it is called as a white gold among the farming community. If the farmer faces anything, their ultimate thing is we have already heard about the farmer suicides. Okay, last 20 years ago. I don't know if you don't know. Also, you go through the Wikipedia, BT cotton. Just you Google it on Wikipedia, you'll get a lot of information. Okay, before the development of BT cotton and what are the production of the BT cotton in India, and before and after the introduction of BT cotton. So whatever the BT cotton which is being developed, that is developed by means of agrobacterium tumefaciens mediated gene transfer. Okay, due to the presence of this technology, we can able to produce the BT cotton. Okay, which is now we are the second most productive country in the world. Earlier, we used to export cotton from other countries. Now we are in a we are importing cotton from. We can export the cotton due to the production of a high amount of cotton that is possible due to the introduction of BT cotton in 2002. Okay, it is the technique which is generally used. So plants are wounded and then inoculated with the agrobacterium rhizogenes. After 2-3 days, plants can be transferred into solid medium with antibiotics such as cefotoxim or vancomycin to kill the bacteria. The hairy roots will be induced within a short period of time, which ranges from 1 week to over a month. Okay, it depends on the species. And decontaminated hairy root cultures can be subcultured on a phytohormone-free medium. It is done, then you can extract the compound from the root system by using the different types of downstream application. Here you can able to see the production of hairy roots. It's a single leaf. There you can use a cotyledon. If you raise those cotyledons on a nutrient medium, okay, this is done by infecting this particular leaf with the agrobacterium rhizogenes. Then only they will produce the hairy roots. Otherwise, they won't produce. Okay. Once you infect that particular plant with the agrobacterium rhizogenes, then that can be done. So, what are the different applications of this hairy root culture? Functional analysis of genes, expressing foreign proteins, production of secondary metabolites, composition of metabolites may also can be changed. You can also regenerate the whole plant, okay, and you can produce compounds which is not found in untransformed roots. That is also one of the beauty. For example, quinine for malaria, chikunine for antibacterial, anti-cancer agent, berberine. All this can be produced by means of hairy root culture. So what are the advantages of hairy root culture or a plant suspension culture? Is fast growth, low doubling time, genetic and biochemical stability in hormone-free media. Okay, so the use of these hairy roots can be used for continuous sources of the production of secondary metabolites. Next comes the third one is the immobilized cell culture. This is the third strategy of production of phytochemicals through plant tissue culture. Okay. What is needed? High cost, slow growth, low yield, instability of selected lines, low resistance of cells, intracellular accumulation. Cells are confined within the reactor system, preventing their entry into the mobile phase which carries the substrate and product. First, report fishes and docus. Okay, it involves two steps. Sorry for interruption. It involves two stages. The first one is optimized for biomass production by suspension culture, and second one is optimized for product formation by immobilized cells. The immobilization, that is, cells are entrapped in a gel behind a semi-permeable membrane. So polymers can be used to entrap cells such as sodium alginate, okay, agar-agar, agarose, carrageenan. Okay, alginate is generally used for polymerization at room temperature, calcium, and also introduction of semi-permeable membrane in between the cells and circulating media, trap cells at cell density. All these things, all these are the different types of cell immobilization, where you can study this cell immobilization technique in enzymes also. If whatever the enzymes which are being produced in the large quantities, all these things are being produced these immobilized cell culture techniques. Advantages: prolonged use of biomass. It increases 2 to 4 times cell separate cell. Prolonged use of biomass, cell density, separate cells from media, downstream processes, growth formation without affecting growth, stable production of secondary metabolites, minimizes fluid viscosity. All these are the different types of advantages. Limitations, as such, decluttering of cell growth, initial biomass must be produced in suspension cultures. Secretion of products into external medium is very imperative. Extracellular degradation of products, introduction of additional diffusion barrier by gel matrix. So the different types of limitations. Good example that is chikungunya and its production. Okay. These are the good examples of how the different types of phytochemicals are being produced. For example, the good example is chikungunya. It is an anthraquinone pigment obtained from Lithospermum erythroxyron. Okay, it is used in healing, antibacterial, anti-inflammatory, okay. Apart from that, this is used in pigments, dyes, cosmetics, and food additives. Choline and alkaline. The pure chikungunya. You see the price, that is US dollars 100. It is also used in the bio lipsticks. Okay, it's a big thing which is used in the cosmetic industries. The biolipistics. Okay, so it's 2 million bees sticks were sold within a few days by Japanese company. Why in vitro production? Food germination, non-availability, plant required 3 to 4 years of noticeable production. I am giving you an example of production of chiconin. Okay, how it is being produced. Apart from that, temperature, pH, light, and oxygen plays an important role. High density, absorption of products, high producing strains, addition of precursors. They are also being uh, enhance the yield of the compound. Biotransformation, that is using bioreactors for production of the important enzymes which are required in a large quantities for the population. Treatment, I already told you about this elicitation process, different types of elicitors. They increase the phytochemicals in the cell suspension cultures. All these are the protocols. I don't think you require all these things. Okay. Important application is chemically they alkalize terpenoids or glycosides. In the pharmaceuticals, flavors, perfume, agrochemical industry, raw materials. Okay. So the production of this phytochemical is multi-billion industry. Okay. 1 kg of vincristine and vinblastine. Have you heard about any time about vincristine and vinblastine? Vinca rosea, Catharanthus roseus, okay, which are can be grown in our gardens. They produce these two alkaloids, vincristine and vinblastine. Okay, the market value of this is very high. Okay. They can be produced in the tissue culture in a large quantity. Apart from that, azadirachtin, berberine, capsaicin, all these are the different types of various phytochemicals which are being produced in large quantities for the ever-growing population. Okay, some can azadirachtin can be used as an insecticide, berberine that can be used as antibacterial, anti-inflammatory, anti-cancerous, capsaicin which is obtained from capsicum. It is generally used for the rheumatic pains. Okay, so the main important theme of this today's presentation is you can produce the phytochemicals in a very large quantities by use of plant tissue culture, okay, in a controlled environment. This industry will going to fetch you high returns. Okay, no doubt the investment is also the one-time investment, but getting a full-time returns forever. Okay, this is one of the important billion industry. In the future, almost all the phytochemicals which are being produced from the plant, you know about the herbal medicines. Herbal medicines are homeopathy or Ayurveda, whatever it may be. All the phytochemicals which are obtained from the plant, they are used in a different system of medicine, but all the things are being used from the plant. So, in the future, no doubt this industry will going to flourish, and they are going to treat the diseases without any side effects. Where in case of allopathy, whatever the chemical synthetic drugs which we are using, you don't know what is the side effect until you use them and experience those things. Okay, I think I'll stop here. If there is any questions, you can. The session is open for discussion.
Girls, good afternoon. If any doubt or any questions, you convey to sir. Any doubts?